let's see what we've got here for the study guide. DNA, its function, genetic information storage consists of deoxyribonucleotides, two types, pyrimidines, purines, know them, okay? A, U, T generally. Purine, well, U is a ribonucleotide, but it's still, sorry, not purine, pyrimidine. A, T, are pyrimidines in the DNA. U is an additional, what am I saying? I'm saying bullshit, sorry. C and T are pyrimidines in the DNA. Just to clarify. C, T, U. A, G. Okay? Remember that. Nucleoside is nitrogenous base, sugar. Nucleotide is nitrogenous base, sugar, phosphate. Two components of nucleotide comprise the backbone, sugar and phosphate. How they oriented? We have 5 to 3 prime, anti-parallel strands. We can read it in a certain direction. Okay, so DNA molecule is directional and 2 strands are anti-parallel. How is the DNA organized? Structurally, double helix, minor major grooves, protein binding, pairs. Oh, come on, by now you should be able to tell this. Which pair is stronger and why? Hydrogen bonds, remember, 3 of them. Chargaff rule percentages. Which molecule we can apply Chargaff rule to? Only double-stranded, right? Not single-stranded. Strands are held together by what? Huh? Hydrogen bonds, yes. If you separate them, you can separate them by heat. It's called denaturation. Why we need to denature DNA? Why denature why it's important that it can be denatured potentially? Huh? Yeah, so we can read it through, right? We can replicate it or transcribe it. Deoxyribo and ribonucleotides, presence or absence of hydroxyl group at 2' location. RNA is unstable. Structural difference. RNA is usually how many strands? 1. It can form internal double-stranded structures like hairpin loops. Can it be denatured? Of course. 4 types of RNA. I'm not going to talk about the function. Just remind you: mRNA, tRNA, rRNA, microRNA, or MI. Relative sizes of genomes. People forget about the numbers. Again, I'm not going to ask you what is the size of a genome of E. coli. Viruses, bacteria, single-celled protozoa, plants and animals. Okay? Fungi is in the ballpark of single-cell eukaryotes, okay? Generally, we can say plants have higher genomes than bacteria, but smaller genomes than mammals. Okay? Phenotype, number of traits, right? Understand what can be a phenotypical trait of a bacteria. Colony color, can it be? Size of the colony, yes. Ability to cause the disease, yes. Ability to be resistant to antibiotic, sure. It's all phenotypical traits. Does that make sense? What defines phenotype? Gene expression at a given time, right? We sometimes say genotype defines phenotype, but let me put it this way: all our genotypes are practically identical to a very large extent, but we look very, very different, all of us. So it's gene expression, not just the sequence. Okay. In which form does cellular DNA exist? Chromosome. Difference between bacterial eukaryotic, bacterial circular, eukaryotic linear. DNA in chromosomes is bound to histones or histone-like proteins in bacteria. Can cellular DNA in the cell in the form other than chromosomal? Eukaryotes. Are there any extra chromosomal DNA? Mitochondria or chloroplasts in bacteria? So they're plasmids. What is the function? Well, kind of self-explanatory. By the way, we never mentioned the function of DNA in mitochondrial eukaryotes, in mitochondrial chloroplasts. Genes are getting expressed. They have their own expression system, so they produce some proteins. Okay. In bacteria, it's what? Conferring traits, right? It can be certain traits on the plasmid that are not essential for the cell. You can take the plasmid out, and the cell is still alive. Okay. Beetle-Tatom experiment. So working hypothesis is that one gene encodes one enzyme. What they did, they disrupted 3 different genes for 3 different enzymes in the pathway, demonstrated that, in fact, yes, each disruption of a given gene disrupted only one enzyme in the pathway, not all this, everything, but just one. Does that make sense? Was hypothesis confirmed initially? Yes. Is that modified now? Yes. Now we say that one gene may encode several polypeptides because of which process? No, why several? Why one gene may encode several polypeptides? It's a process between the transcription and translation. Splicing, because of the alternative splicing, right? Transcription, RNA binds to the promoter, okay, which is ahead, upstream of the gene, starts the transcription. How many strands are transcribed? 1. How many strands are needed for transcription? 2. DNA must be double-stranded. Does that make sense? Good. The product of transcription is mRNA, single-stranded molecule. I wanted to ask something else. Brain freeze. 3 stages. Oh, yeah. 3 stages: initiation, elongation, termination. Enzyme that facilitates the process. Which polymerase? Which polymerase? RNA polymerase, period. Okay? Well, in eukaryotes there are 3 of them actually, but let's not get into that. Promoter, direction. 3 to 5 prime, termination, terminal repeats. Repetitive sequences on the DNA that stall the RNA polymerase. Okay? I'm telling you what you should concentrate on. Splicing. What are we cutting out? Introns. What do we leave? Exons. Which organisms do splicing? Eukaryotic. Viruses can do it too. Which DNA occupies more space in genome? Non-coding. The possible function? Regulatory. Alternative splicing, when you combine different exons together. Cool? Genetic code work. 3 nucleotides form 1 codon. We have 64 codons, 3 of them are stop, 61 encode for amino acids. 1 amino acid may be encoded by more than 1 codon. The evolutionary advantage is that those particular amino acids are less prone to change as the result of mutation, so you have higher chances of silent mutation. How many amino acids one codon, one given codon, how many amino acids it encodes? 1. How many amino acids are in, well, I shouldn't say in the protein, I should say in a protein. How many? Possible, 21. Okay. Where does the translation happen? Ribosomes. I specifically say non-membrane bound because I don't want to focus on eukaryotes too much and talk about endoplasmic reticulum. They consist of what 2 types of molecules? Small unit and large unit, but molecular level are RNA and protein. Yes, RNA and protein. Bacterial eukaryotic. Again, the answer is there are some. No, seriously. I hate to make people memorize numbers. A little digression. Somebody told me that here in high school, in chemistry class, students have to memorize the periodic table. When I was told this, like a year ago something. Maybe in some schools over, they made you do that? I went, so 5 years of chemistry in college. This thought never occurred to anyone. We never had a single question about memorizing the thing. Because why memorize something that either is on the wall or can be easily Googled? If you remember the stuff but have no, it's like bar mitzvah. We discovered bar mitzvah a year ago, so we're now familiar with the whole concept of bar mitzvah. When poor kids have to memorize and read the fragment of Torah in Hebrew that they don't know, written in Hebrew, they read an hour of a language they do not understand. So it's, well, okay, that's religious thing. I get it. Chemistry is not a religion. You can understand the goddamn periodic table. Why memorize it? Same here. As long as you know, and it's like ingrained in your mind, that bacterial and eukaryotic ribosomes are very different. Very different. That's important. He has bacterial 70S, eukaryotic 80S. If you're going to work on them, it's fine. For your patients, I bet they don't give a shit about the numbers. Okay, as long as you know why tetracycline works on microbes, on bacteria, and why it doesn't kill them, you're good. How amino acids are transported in the cell? What moves amino acids in the cell? tRNA. Order of assembly of translation, initiation complex. mRNA binds small subunit first, right? Small subunit of eukaryotic binds to the 5' cap, prokaryotic to the Shine-Dalgarno sequence, and then it binds tRNA to remember which first amino acid. Methionine. MET, methionine. The reason, because start codon, AUG, is also methionine codon. It's always methionine. That's the reason why. And then you have the large subunit that sits on top of that stuff, and you get your initiation complex done. Describe the process of protein synthesis. Remember that little play that we've done? If you can recollect it, that's it. So A site, B site, E site. What are the functions? Where does tRNA go? Okay. The incoming goes into A site. The growing polypeptide chain is in the B site. Okay. Where does it happen in proeukaryotes? Already mentioned the in prokaryotes cytoplasm, in eukaryotes cytoplasm and endoplasmic reticulum. That's where the ribosomes are. What is transferred to what? Yes, polypeptide chain to the incoming amino acid. How does the ribosome know when to stop? You have stop codons. What if there is a special sequence that is called selenocysteine insertion sequence after the stop codon? It signals this stop codon actually is not a stop codon. Keep going. It's an insertion of selenocysteine, the 21st amino acid. Okay, I usually compare this selenocysteine insertion sequence. You drive down the road, there's a stop sign. So you want to stop. There's a police officer behind the stop sign that shows you, come on, move on, come on, let's go. Does that make sense? And you, okay, yeah, police officer tells me, and you drive forward. Okay. Prokaryotic gene expression. That picture. Prokaryotic is simultaneous. Eukaryotic is separated in time and space. Prokaryotic mRNA is not modified. Eukaryotic is splicing, capping. Prokaryotic polycistronic. Eukaryotic monocistronic. So kind of where the transcripts, transcript is in the nucleus, in eukaryotes, and then RNA has to be transported in the cytoplasm and endoplasmic reticulum. Does that make sense? Purpose of DNA replication. We pass genetic information, right, to the next generation. At which step does it happen? What do you think? Before it. Just before cell division. In eukaryotes, it's going to be S phase between G1 and G2. In prokaryotes, before cell is ready to divide, like bacterial cell is about to divide, it replicates DNA, and there we go, we have binary fission. Where does it happen? In eukaryotes, where does it happen? Nucleus, yes. In prokaryotes, just wherever DNA is. Why two strands? In case one damaged, yes, to protect from damage. Semi-conservative replication means that each new double-stranded molecule consists of one old and one new individual strands. If you don't mind, I'll skip this. There's a table. I refer to the slide. I usually don't do this, but it's just easier. I gave you the reference slide number 15 with a table with the enzymes that participate in the DNA replication. I told you the questions, you saw the questions. This enzyme unzips double-strand DNA molecule, and you pick one answer out of 4. Does that make sense? That's going to be the type of questions that you're going to see about the enzymes. Replication starts at the origin, which has a lot of which pairs? ATs, because they are easier to break. What is the direction of reading the strand? 3 to 5. Untangling and unzipping the primers, the RNA primers required to start the replication, which is selective. Leading, lagging, leading, uninterrupted synthesis, lagging, interrupted synthesis. Okazaki fragments, short fragments that are synthesized in the lagging strand, because DNA polymerase that's on the lagging strand has to like move, sort of jump backwards all the time. Why? Because of the reading direction, 3 to 5. How they're getting stitched together. Ligase, what are concatenated chromosomes? As far as I know, when they're separated, this is concatenated, this is called catenated. Okay, two circular chromosomes locked into each other, right? And then topoisomerase separates them. Mutations, changes in the DNA or RNA. Can protein be mutated? No. No. What can cause the mutation? Can be chemical. That's what I refer to the table. So we have nitrous acid or different nitrous compounds. They change nucleotides, modify nucleotides. So it's going to be point mutation without frame shift. We have nucleoside analogs that also change the sequence without shifting the reading frame. We have intercalating agents. That's either insertion or deletion, so frame shift. And we have radiation that we talked about. Non-ionizing causes the dimers, okay, thymine dimers. Can be point, can be insertion, can be deletion. Ionizing strand breaks, which can cause the mutation after the repair. It's the result of repair. Does that make sense? Crap. Missense, nonsense, silent mutations, frame shift, reading frame. I separated these two things here. Separated these two just because, so you have a little bit more space. Okay. Frame shift, either insertion or deletion of the nucleotide or two. Which one's worse? We talked about this whole thing, okay? Which one's worse? Ionizing, because it can cause double strand break. Does that make sense? Cell repair, the excision repair, it happens in the dark, and photolyase, that happens in the light. So excision, cut out the damaged portion, fill it up. Photolyase, just break down the bond between two thymines. So that's slide whatever number, we just have an explanation. Ames test. Detecting mutagen, oxotrophic microorganism requires a certain growth factor. If the chemical is mutagenic during Ames test, oxotrophic mutant will revert to the wild type, okay, being non-oxotrophic. Kind of medium, we use medium that lacks that essential nutrient. And microbe is oxotrophic for, how do we call organisms that return to their original phenotype? Revertants. 3 mechanisms. Transformation, transduction, and conjugation. Okay? Importance. Lack of sexual reproduction, so they have it to increase the diversity. Eukaryotes, sex. Do prokaryotes have this option? No. Does that make sense? What I'm saying? What is transformation? Griffith experiment. Live microbe acquires free DNA from the environment. Importance of this process in the public health. Antibiotic resistance. Biotechnology applications. Inserting necessary genes into the certain microbe. Okay, so it can produce something. What is transduction? Phages, holding fragments of the bacterial DNA between the cells. Lytic phages do general, be able to explain what's going on. The fragment of the bacterial DNA is directly packaged into the phage particle. Specialized transduction is done by lysogenic phages. And a fragment of bacterial DNA is attached to the phage DNA. What happens to the donor cell during the transduction, either general or specialized? It dies. Conjugation, bacterial sex, exchange of DNA between two live cells. Remember? Live cells, they don't die afterwards. Plasmid, fertility, external appendages, pilus, two conjugating cells. Do they have to be of the same species? Do they? No, they don't. Again, all this genetic exchange of genetic information important for public health is antibiotic resistance and virulence, different virulence factors. HFR cells, when fertility plasmid inserts itself into the chromosome. When it is excised, it often carries a fragment of the bacterial chromosome, so it becomes F prime. Okay? Is it possible to transfer the bacterial chromosome during conjugation? No, because it's too big. And then, but we can map the genome based on the time that is required for a certain gene to enter the another cell during conjugation. What's operon? Several structural genes that are used for the same sort of metabolic process, same function. Promoter, you know what promoter is. Regulator is the sequence that repressor protein binds to. So structural genes comprise operon. Understand the difference between inducible and repressible. Inducible is regulated by the substrate of the metabolic pathway. Repressible is by the product. So type of enzymes, they usually encode what I mean by that. Enzymes that will convert the substrate. Repressible will make enzymes that synthesize the product. Riboswitch, small sequence in the RNA that responds to the small molecules, regulates transcription and translation. Small molecules bind to the hairpin loop. And hairpin loops can be found in what molecule? RNA. There you go. Metabolism, catabolism, anabolism. Know the definitions. Classification of organism based on the source of energy. Remember that table? Know that table. It's a slide for metabolic. So yes, you can get a question. You probably, has everybody got the quiz yesterday, not today? Yeah? You have seen the quiz? No? You didn't? No? The quiz. I said that I'm going to give quiz today, but a lot of people decided to take it yesterday. It's fine. Yeah, you can take it. But the point is, there's questions like chemoautotrophs and then definition. That is the question that you may see. My photoautotrophs are doing this, this, and that. Okay? Photoautotrophs are plants and algae. Most microbes, like most bacteria, practically all fungi, mammals, they are what? What do they use as a source of energy? Chemicals. So chemo. What do they take carbon? Organic or inorganic? Organic. Chemo heterotrophs. Does that make sense? I didn't try to scream at you here, just a typo. Oxidation reduction. Be comfortable with the terms electron transfer. It's actually very exhaustive. Function of electrons in the cell. High energy electrons, they actually kind of transfer energy from one molecule to another. Electrons are transferred by the electron transporters. Function of ATP, energy currency. This, I give you the reaction, and you tell me if it's exergonic or endergonic, anabolic or catabolic. For instance, protein synthesis requires enormous amount of ATP. Protein synthesis requires enormous amount of ATP. Characterize protein synthesis based from the standpoint of exergonic, endergonic, anabolic, catabolic. If it requires energy, it's what? Endergonic, and its synthesis, anabolic. Make sense? Good. It's not 100% effective. Not a single chemical reaction is 100% effective. I mean, energy conversion. Enzymes. Major features, specificity. They're not used in the reaction. They alter rate but not ratio. Substrate and product. Please remember that basic idea. Okay. Substrate binds to the active site, converted into the product. Exoenzymes. Location. Remember we discussed that exoenzymes are frequently the virulence factors because they may damage the tissues. Exo, outside, right? Regulation of enzyme activity. Inhibition, competitive, non-competitive. Allosteric activation. Remember that non-competitive inhibition and allosteric inhibition are practically synonyms, yes. Feedback, negative feedback. We can observe negative feedback. Where do the inhibitors bind? To the active site or to the regulatory site? Glycolysis. You went through this so many times. Things that we discussed in the class. I will not go beyond that. But I expect you to know that. So don't try to convince me that CO2 is produced during glycolysis. It is not. Okay. Of course, I'm not going to ask you like names of the enzymes and so on and so forth. I don't really care much about that. ATP yield, what else produced during glycolysis? No, any carbon dioxide. If you answer each and every question on the study guide, you should be fine. I mean, correctly answer. Come on, they're simple. It is oxidized to acetyl-CoA, to citric oxalic acid, 4 citric acid, 6. Okay, so you have 2 carbons in acetyl-CoA, 4 carbons in oxalic acid, 6 carbons in a citric acid. Okay? Citric acid oxidized to isocitric, then alpha-ketoglutaric oxidized to succinyl-CoA. Product functional importance. Okay. What else is produced in the Krebs cycle? What is produced in the Krebs cycle? Is CO2 produced? Yes, during Krebs cycle, CO2 is produced. What else? NADH, FADH2, and ATP. What should I say, the most important molecules there. NADH and FADH2. Why? What do they carry? Electrons. So that's what we want. Does that make sense? Awesome. Not a lot of ATP. What happens to the remnants of the original glucose molecule during Krebs cycle? During Krebs cycle, glucose is completely oxidized. Krebs cycle finishes the oxidation of glucose. How many Krebs cycles? 2 to oxidize 1 glucose molecule. Summary outcome, including transition stage. Know the molecules that are produced. What are the uses? Oxaloacetate and alpha-ketoglutarate, amino acid and nucleotide synthesis, citric acid, lipid synthesis, succinyl-CoA, synthesis of things like chlorophylls and heme and other porphyrin molecules. Kind of list it all here. Electron transport chain. I don't want to recite it again. Electrons give energy. Energy is used to pump hydrogens. Hydrogen diffuse back. Energy of the diffusion is used for ATP synthesis. Electrons go to the oxygen. We call it final electron acceptor. That's protons, ATP synthase, proton diffusion. Fermentation occurs when what? Microbes cannot use oxygen, right? That's the point. Where do electrons end up in fermentation? Organic molecule, final electron acceptor. In anaerobic respiration, final electron acceptor is inorganic molecule other than oxygen. Why organisms cannot carry out aerobic respiration? Either there's no oxygen, they don't have genes for electron transport chain, or they don't have genes for the enzymes that can neutralize reactive oxygen species. Okay. Fermenting organisms lack electron transport chain. Okay. Aerobic and anaerobic respiration have different electron transport chain. Two types of fermentation, acidic and alcoholic. Final electron acceptor for acidic is pyruvate, for alcoholic acetaldehyde. Lipids, lipid metabolism, glycerol goes into the glycolysis, and free fatty acids go through beta oxidation, become acetyl-CoA, which enters the Krebs cycle. Amino acid, oxidative deamination or transamination, they may enter the Krebs cycle or become pyruvate. Evolutionary purpose of photosynthesis, just discussed today. Two types of light-dependent, light-independent, photopigments, where you can find them. Chloroplast, we discussed different colors, why different pigments to absorb as much light as possible. Light-dependent stage, photosystem 2, photosystem 1. What are the steps? What are the products? Where do electrons go? From water. And then you have detailed questions, okay? ATP produced by photosystem 2 and ATPH for system 1. It is oxidized. Can other molecules be electron donors? Yes. Anoxygenic. And a couple of things. Light independent reaction. Carbohydrate. Okay. Calvin-Benson. Fixation of CO2. ATP and ADPH. ATP is used to phosphorylate intermediates, and ADPH donates electrons. Okay?